interested in immunotherapy for multiple myeloma, as well as other novel drugs and using MRD as an endpoint for clinical trials. So some of the research that we're participating in at our institution includes chimeric antigen receptor T cells, or CAR T cells, as well as bispecific T cell engagers, commonly known as BiTEs. We're also very interested in understanding if novel immunotherapy combinations, for example, daratumumab up front, can make things better for patients in the long term. In addition, we're excited to launch soon a trial called the ARIGA trial, which is being run by Janssen and is going to use MRD as an endpoint. That is, patients who are in CR or VGPR after transplant but are still MRD positive by a next generation sequencing technology will be randomized to get either daratumumab and lenalidomide or lenalidomide alone. And we're really excited because the endpoint of this trial is is MRD negativity at 36 months, which is a new kind of endpoint, and I'm happy to be a part of that. A lot of people want to know how we can use MRD. I kind of always make the joke that MRD is like wedding china. It's great to have, but you don't know what to do with it, right? We know a couple of things about MRD. Being MRD negative is being better than MRD positive, and you can correlate progression-free survival with MRD outcomes, so that's good. But how do you get there? Do we use MRD as a decision-making point? And that we don't know yet, and we don't know if it's overall survival going to be a pertinent endpoint. Because of that, it's imperative for us to do clinical trials where we use MRD to say, hey, we're going to stop lenalidomide maintenance, or hey, we're going to add something because you're negative or positive, and use that to guide our decisions and see what the data is. And then we'll be able to tell our patients, hey, we got this sample, you're MRD positive or negative, and now I can do something about that.